The Paskers is one type of the Paskers of molecular basis of inheritance, and today I will discuss about concept of DNA packaging. Okay, first we discuss the heading part. What is DNA packaging? For the meaning of packaging, packaging means we know that when some goods are we buy from the market and collect them in one place, that it was packed by some pockets or bags, polythenes. This is packed some goods by the polythene and these things. Okay, at this similar type, the DNA was packaged by the histone protein. Okay, before going this concept, we know that positive charge always likes negative charge, and positive charge always dislike negative charge, so that the DNA always negative. It was comes in the exam, it will come in the exam as a short question also. DNA is a negative particle charge, but when we added a positive type of charge with DNA, then it can be packed by this positive charge, so that it happening in two types. That is, we know that two type of cell present in our world. That is, one is prokaryotic cell and another is eukaryotic cell. The prokaryotic cell is a primitive type of cell, so that we always consider the eukaryotic cell, because eukaryotic cell is completely based upon the all organelles. That means in eukaryotic cell the all organelles are present. That means that is mitochondria and ribosome, endoplasmic reticulum, microtubules, all part present in the eukaryotic cell, so that eukaryotic cell is much more complex than the prokaryotic cell. Okay, so that we discuss how the DNA is packed in eukaryotic cell. DNA packed by the help of histone protein. Histone protein is completely complexed by the positive charge, because it is one type of amino acid, and this amino acid is residued by lysine and arginine. Arginine and lysine present in histone protein. Histone protein is 5 types. Okay, H1, H2A, H2B, H2C, H3, H4. 5 histone protein are present. In this 5 histone protein, it is further divided into 2 types. One is linker histone protein and another is core histone protein. The H1 is only one, the linker histone protein, but H2A, H2B, H2C, H3 and H4 are core protein. What is linker and what is core, and how the DNA circulates itself inside the histone protein, we know this from the structure. Okay, this is the core. The ball size or circular size structure is the core protein. In this core protein, the H2A, H2B, H2C and H4 core protein are present. It is present pairwise. That means the H2A protein, 2 H2A protein is present, 2 H2B, 2 H2C, 2 H4 protein is present. That means 8 core histone protein is equal to 1 core of histone. Here 8 core protein is present, but H1 is only one protein which is called as linker protein. Why called H1 is the linker protein? Because H1 linked the two end of the DNA. The two end of the DNA was joined by this linker protein. A linker protein links the two end of the DNA. This is the DNA that circulates itself in the core protein, and linker help to link the two end of the DNA. Okay, in when the DNA linked around or circulate around the core protein, it helps 146 base pair. 146 base pair needed for completing the structure of histone protein. That means 146 base pair is to form the linker and histone protein as packaging. In a packaging, 146 DNA base pair is used, so that we used 146 base pair, and in one cycle 20 base pair is used. One cycle 20 base pair is used. It is most so type question that comes in the exam. Most important this part, so that it was the concept about histone protein. Histone is the positive charge species, but DNA is the negative charge species, so that the histone protein is attracted by the DNA. The DNA is attracted by the histone protein because DNA is negative charge particle, but the histone protein is positively charged particle. When histone protein are linked together one by one and a chain type structure is formed, then it is called as chromatin. Okay, this is called as chromatin. When chromatin are loosely present, this chromatin is called as euchromatin. When chromatin are seen densely, that means this type of chromatin is seen, then this chromatin is called as heterochromatin. Okay, when it is seen loosely, it is called as histone euchromatin histone protein euchromatin, and when it is seen as densely, then it is called as heterochromatin. This was about the concept of DNA packaging. In this way, the DNA is packed and packed and packed. Here the DNA is packed, DNA is packed, DNA. Here the DNA is packed by the help of histone protein, and histone protein further complex to form chromatin, chromatin then complex to form chromosome. This way the DNA is packed by the help of histone protein. Thank you all of you. This is my crash course. Today is the third day of my crash course. Please like, subscribe and share my channel, and see every video in every day. Thank you.